All right, class, welcome to week 2, lab 2 of Bio 105. It's great to have you here. I'm just going to go through a couple housekeeping things. I'm putting a link to all our recorded labs right here on kind of your first getting started module. There's a link to all the recorded labs. There's a link to my office hours and our normal live class Zoom link. If any of you guys added the class late, I'll still allow you to make up some assignments. Some people weren't able to complete all the assignments because they added it late. So feel free to, I think you're able to keep doing these assignments or uploading or submitting a worksheet. Your lab one from last week was due on Sunday. I'll try to get those graded by this weekend. So always expect me to grade your work within 7 calendar days. And I'll try to stay on top of that as well. The only other thing I want to point out is then, so each week you have a different lab worksheet due, and then each week you'll have a quiz on the previous week's lab. So what that means is that this Sunday, September 5th, you have your lab 1 quiz. It's just 10 points. It'll be a lot of conversion problems, mostly multiple choice. There might be some, like maybe 1 fill in the blank, about 10 questions. And that's remotely proctored, so that'll use Proctorio, so you can't use any notes. So that's worth 10 points. How I grade your worksheets is I will grade your worksheets a lot of times on completion. If I see that you really are struggling with some, I might take a point or 2 off. But if I see that you put in some effort, I usually grade them on completion. If you ever want to go over a worksheet or go over the answers, let me know and we can use this lab time to do that. A good way might be to go over the answers if you want to on your worksheet during lab time before you might take the quiz for that week's lab, if that makes sense. And then today is all on microscopes. So there is, just like last week, there's a lab guide and a worksheet. And if you can open up both at the same time, or if you go back and forth between the two, your lab guide will guide you through the questions on the worksheet. And just like if we were in person, I'm going to introduce the topic of microscopes. We'll probably spend maybe just 20 minutes together, half an hour, maybe longer if you have questions, and then I'll let you guys have time to work through the worksheet on your own using the lab guide as a resource. And again, if you have questions, come see me in my office hours tomorrow or send me a message and we can set up another time to meet. So by this Sunday, you'll have the lab 2 worksheet. You can submit that here. As well as the quiz from lab 1 to do. And then the quiz on lab 2, which will be on microscopes, won't be due until the following Sunday. So I just wanted to make sure we're all on the same page with that. We are going to keep the same format for lab, a worksheet and a quiz due every week until our lab final. So you guys won't have any other kind of hidden assignments. You'll just have a quiz and a worksheet due every week with our lab class time recorded. And then at the end of the semester, you'll have a cumulative lab final based on kind of all of the topics up until that point. So I hope that's really straightforward with you guys. It's something you can handle, keep track of on your own. I always post the lab information on Monday morning. So if you ever want to get started on working through the labs, feel free to do that because I think a lot of this you can kind of work a little bit on your own. Okay, so this is the lab guide to the introduction to microscopy, which is the use of microscopes. And what I'm just going to kind of go back and forth. I'm going to go back and forth here. I'm just going to see something here. All right, I'm going to go back and forth between your worksheet and the lab guide. So you're looking at the worksheet right now. The learning objectives, you should become familiar with the names and functions of the different microscope parts, know how to safely handle them and care with them, know how to improve your technical skills. So we'll go over a lot of these the best we can in a virtual setting. The introduction to a microscope. So the light microscope is what we are familiar with in lab, is an enormous importance in biology because it has the ability to see something very zoomed in. When carrying it, you should always put one hand on the base and one hand on the arm. It consists of a stand where there's a stage. I'm going to go over all of these parts by looking at a picture. And here's a picture of the microscope. And your first kind of part of the worksheet is you have to identify, locate, and know the function of all of these following parts. And you have to label them based on the pictures shown in the lab guide. And the lab guide, so the first thing you can do is you can watch a microscope tutorial of this YouTube video, which I encourage you guys to do on your own. And then this is what the microscope parts are. So when you're carrying the microscope, you should put one hand underneath the base and hold it by the arm because they're very heavy. And as you're looking into the microscope, you'll see this black stage down here. This stage is what you'll place in kind of the glass microscope slide into these slide holder or slide clips. And these little rectangle kind of specimen slides are what we kind of dye our specimens or our subjects. And then we take those glass slides and place them onto this stage. What you will do then is you'll look through the ocular lens. So you'll put both your eyes and you'll look through the ocular lens down at this sample. How you're able to see the sample is that light is coming up through the light source and it's shining through this hole. And right where this hole is where you'll want to kind of arrange your sample so that what you're looking at is you're looking at light shining through the sample, and the sample will always be dyed, usually purple or blue. So when you're looking at your sample, as you're looking through these ocular lenses are already about 10 times magnified. So you're looking at something about 10 times magnified by looking through the ocular lenses. And then these objective lenses are all of these kind of different colors. There's a red, a yellow, a blue, and a white, and they have different lengths to them. They're on a resolving nose piece because you can rotate the objective lenses. And each of these objective lenses stands for a different magnification power. So for example, the red or the shortest is called your scanning power objective lens. And that's for just, you try to start with that objective lens first, because that gives you the least zoomed in power. So as you're trying to identify your sample, you want to start, it's called with the scanning power because it's not so much zoomed in. So you're better able to see the sample before you start to zoom in. You'll use the coarse adjustment knob, which is right here. So you kind of have these two adjustment knobs on the side, and that's what you'll turn that will allow you to kind of zoom in or zoom out on your sample to focus what you're looking at. And you'll use the coarse adjustment knob when you're first trying to locate your sample. So these objective lenses have different magnification powers as well. The scanning and then going up to the yellow, the blue, and the white, they all have an increase as they get longer. The longer they are, that means more zoomed in they are. And we'll go through those. And as well as the tutorial in this video, which I encourage you to watch right after we're done meeting, will take you through these different parts of the objective lens. So you'll use a different objective lens to zoom in more on the sample. You have the stage clips, the condenser, and the iris diaphragm level. So the light comes up from the light source. So you'll turn on the power switch. The light comes up, and then the diaphragm lever and the condenser just control how much light travels up through your sample that you're looking at. The X and Y axis knob, these just help to arrange the sample to kind of lie right on top of the light source and right underneath the objective lens that you're using. So the whole point of a microscope is to kind of focus in on your sample that you're seeing so that whatever you're looking at, a cell, a piece of a cell, you can zoom in on it and see it much better than you would if you were just looking at it with the naked eye. So your first part in your worksheet is to identify all the parts of the microscope. And I'm going to kind of go back and forth to and from the worksheet to kind of walk you through it. The next part of your worksheet is you have to calculate total magnification. There's the tutorial video that you can watch that will help you with this, but I will also talk you through about how we calculate total magnification. And in your worksheet, so the scanning power objective lens, this was the red color, the shortest color objective lens. And then you had a low power, a high power, and a high dry power. I think if I'm looking correctly, going up, you have a yellow, blue, and white. So they kind of give them different colors of the microscope just to better identify the objective lenses themselves. And in order to calculate total magnification, all you do is you multiply the objective lens power by your ocular lens magnification. The ocular lens are what, again, you look through in the microscope. And remember I said that when you look through those, you're looking at the sample already at 10 times magnified. So these will be the same for all the ocular lens. You are always looking at something 10 times magnified. And what you'll do is you'll multiply by the magnification of the objective lens. And you'll have to go through and watch that tutorial. You can look at reference guide page number 4 to get the objective lens magnification of each of these. I'll help you with one of them. I'll help you with the first one. The scanning, the kind of the lowest zoomed in objective has a magnification of 4x. What that means is that when you're using this objective lens magnification, it'll zoom in on something 4 times. To get the total magnification of the sample, you take the 4 times the magnification power of the objective lens, and you multiply that times the magnification power of the ocular lens. So the total magnification, if you're using the scanning, the red objective kind of structure, you'll be looking at a structure 40 times magnified. The other one we give you is the high and dry power is a 60 times magnification lens, and you just multiply that by 10. So when you're using this objective lens, you're looking at something 600 times magnified. So that's how you do kind of this table, and I'll let you go and figure out the yellow and blue, and you can watch that tutorial video from Lab Guide 2 to figure out those. But again, all you're doing is you're multiplying this column by 10 to get the total magnification power. Using the compound light microscope, the compound light microscope should be treated as a very fragile. The objectives for your microscopes can cost $200 or more. For this part, see Lab Guide page 5, and this kind of takes you through about the steps in how to use it. You'll turn it on. You'll start with the lowest objective power. You'll use the X and Y axis knobs to position the slide so that the portion of the slide is under the 4x objective lens. You'll turn the off switch on. So this takes you through the steps in how to use a microscope. I'm going to go through, and it said to go through, I'm just going to share you the lab guide. So again, you can work through the lab guide and the worksheet at the same time. There's nothing to complete or answer in that section. So you can kind of just read through that section. Part 4 is calculating the field of view. In the lab, we would use a slide with a tiny ruler scale to calculate the diameter of the field of view, and instead you will visit the website indicated in your lab to complete a simulation for calculating a field of view. What we're talking about here is we're going to try to calculate the distance or the measurement of the field of view that you're looking at. And you guys will use your worksheet, this website, to calculate this field of view using a microscope and a simulation. So I will let you guys visit this lab website. You'll click on the image of the microscope to begin the simulation. You'll read the information provided. You'll follow instructions under the procedure. You'll measure the field of view, and you can kind of convert it into millimeters, and it kind of takes you through this. So I'm going to let you guys use this, kind of work through this on your own. But if you have questions on this, let me know, but you'll have to use a procedure found in this simulation. Table 2, the field view calculations. You'll be able to complete this table when you stay on the website and click next through the simulations. And again, all instructions are shown here on how to calculate those field of views. Estimating an object size. So now you'll estimate the size of an object that you're looking at, and you'll go through these lab pages in the lab guide. You'll read the information on estimating the object size. The activity will show you the application for the field of view measurements that you obtained from the previous part, and you'll use this skill for the remainder of this term to estimate microscopic specimen sizes. You'll use the instructions on page 6. Email if you have any questions with the STEMI simulations. So that takes you through that part. Part view will be using a prepared slide. You'll read the information at the top of page 7 on your lab worksheet. So let's move on to page 7. What you're going to be doing is prepared slides come with mounted items ready to use. So the prepared slides are what we would be using in lab if we were looking at skin cells or a bacteria. Often a chemical stain is added to make the features more visible. Always be sure to know the function of each part labeled and include the statement prepared slide along with the title of your drawing. So you'll be making some drawings. You'll include a title, total magnification, and you must also use a size scale or an approximate length and approximate width. So here we have total magnification. This is an example of a paramecium, a bacteria, and a generalized kind of length and width of that bacteria. So this is what you'll be looking at, a prepared slide. So I'm going to go back to the guide here. Here's a young sea star at 400 times total magnification. So the lab guide gives you the title of the drawing that you'll use, as well as the total magnification that you'll add. And you'll just sketch this specimen on the first drawing page in your lab document. Here's a sample drawing page for a prepared slide. You have a title, the name, the date, the total magnification at the bottom, and you'll be doing a drawing of this prepared slide, which we're giving a picture of. So we give you the total magnification and the title, and then just include your name. Creating a wet mount slide. This is a slide that the user makes of a sample in a laboratory setting, like putting some drops of pond water onto a clean slide to look at for microbes. I've also done a sample of your skin, the inside of your mouth. We'll be looking at tutorial videos, and here are 2 additional videos to reference for this procedure for creating a wet mount slide. And let me go back to your worksheet. You'll also be looking at the letter E. You'll be looking at a video for the letter E demonstration, and you're going to be answering some questions about what the letter E looks like underneath a microscope. And what this is getting you to answer is, when you write the letter E down on a piece of paper, it normally looks a certain way, but when you look at the letter E underneath a microscope, it's going to look different. And what this is trying to get you to understand is when you're looking at microscopic specimens underneath a microscope, they might be backward or inverted, and you'll be able to tell that when you look through the letter E slide demonstration. Viewing the prepared slide, we kind of talked about this already. Creating a wet mount slide, you'll watch this video on how to create a wet mount slide. You look at those examples, which I showed you in the lab guide. The checkout procedures, these are just more for informational purposes. If we were, together in person, you would do the checkout. And this is the drawing page where you would draw those example specimens. So that's it for this lab. It's a little bit simpler. It's kind of just to give you an introduction on how to use a microscope. I'm going to go ahead and go back to your lab guide to make sure we got through everything. Again, sample drawing page for a wet mount slide. It just gives you pictures for that, reminders and tips. So I'm going to go ahead and stop the recording as this takes us through kind of an introduction for this lab.